then one thing about mine is like apparently there's just like crisis on it, so I was like, why I would not find it not going but because I was just like, I got and like students who got higher amount than me will like invest things, had a worse GPA, and worse colleges. Really? Like I don't want to like be rude, but at the same time, really? Why do you have such a large jar of peanut butter? Like don't even cry because it's kind of my lunch.
So we started on today. We went over those papers before P days, right? We did that paper, you guys each gave groups. Now we're going to talk about what we've learned since that initial paper RNA came out. They were proposing a couple different mechanisms and they didn't know, but the summary of that paper basically was they proposed two different ways that they envisioned that this double-stranded RNA could shut down gene expression. The double-stranded RNA, first of all, they demonstrated it's enzymatic. And the experiment they ran to show that is, if I put 60,000 molecules into the organism, and that organism then divides into 1,000 cells, the maximum number you could have is 60 in each cell, right? And if we know that each cell makes 1,000 an hour, 1,000 messenger RNAs an hour, the only way that those 60 could keep knocking that gene out is to be an enzymatic process, right? 60 to knock out 1,000 an hour. So they ran some simple experiments. They said, well, if I use the sequence, if I make double-stranded RNA to be the sequence of the messenger RNA, does that knock out gene expression? Yes. If the double-stranded RNA is the sequence of introns, so if it's the sequence of pre-RNA, sequences not found here, which means that are cut out, does that shut down gene expression? And no, that didn't. And then, if I use double-stranded RNA sequences that are the sequences in the promoter, can I shut down gene expression? And the answer was no, that did not shut down gene expression. So they suggested that the double-stranded RNAs, those small RNAs that were shutting down gene expression through this process, were working at this stage. And this is phenomenal, because up to then, at the time they published this, we knew everything we needed to know about messenger RNA being degraded. If it lost its cap, the messenger RNA would be degraded. If it lost its poly A tail, the messenger RNA would be degraded. The way to destroy a messenger RNA was to knock those things off. So somehow, the CRAS, these double-stranded RNAs, are the mechanism that you decap, you depoly A, take the poly A tail off of a messenger RNA. This may be the process through which that happens. This is the machine that actually does it. They also considered the possibility that the double-stranded RNAs might somehow go to the DNA and affect DNA compaction or affect transcription, this process here. They didn't know, but they didn't find evidence for that. And today, I'll actually talk about the machines that actually do the work, that use the double-stranded RNAs to perform the work. So, get this up here. So these double-stranded RNAs, people, at various times, people would purify, this back when I was in grad school, ancient, long time ago. They'd purify nuclei, they'd break the nuclei open and run gels and separate out the nucleic acids. So on a gel, what you get in this situation is, and we're going to run a very long gel, and probably 3% agarose, or 5% agarose. So it's really thick, and it takes DNA a long time to work its way through. You have a huge, huge long gel, so you can separate things out. And down here, you have really small nucleotides. And up here, you have chromosomes, right? And you can can separate things out. It's actually really hard to get a chromosome to go into a gel. I won't talk about the mechanism. There's special ways to get chromosomes to run into a gel. And when I say very small, we're talking 20 nucleotides. Okay? And there's stuff all along the way. It's just a smear that runs all the way down. And there are bands that are really intense at various spots. Some spots are more intense than others. It turns out these really small ones are the brightest. Most of the nucleotides are in these really small ones. These, we can actually cut these out and sequence them, and those are ribosomal RNA sequences. Ribosomal RNAs, the RNA that is part of the ribosome that does translation, right? So we make so much of those, and then all the transfer RNA genes, we could identify those as distinct in the gel. So we know that these are chromosomes. So we didn't know about these microRNA things. So for decades, people knew that these things existed. Since they didn't know that microRNAs existed, what did they explain these things as? You should look to this expanded central dogma over here.
Just random degraded stuff.
Yeah. Yeah. It's these degraded bits of messenger RNA or transfer RNA or whatever. You've caught things that are in the process of being broken down into nucleotides, NTs. So this is junk. It's garbage. It's clogging up the nucleus, whatever. So what these guys found is actually some of these guys are functional. Some of these small RNAs are the machine that actually performs the degradation of the messenger RNAs. What happens is there is a double-stranded RNA, and this might be 1,000 base pairs long if we put this in here. And there's a machine that comes in and will bind to a small portion of this. And to the best of my knowledge, where it chooses to bind is random. It's probably not just as random inside the cell, but anyway, for our purposes, we'll say it's random. This machine cuts here, and we'll say that goes away and is destroyed. And it cuts here, and this goes away and is destroyed. And the machine holds onto this small little bit. This small little bit, depending who you talk to, they might say it's 40 nucleotides, 40 base pairs long. It might be 25 base pairs long. And then it gets trimmed and processed in certain ways. So that's represented here. I guess this shows 21 nucleotides, 21 base pairs long. So these things go by different names. Some people refer to these as silencing RNAs. And there's a company called Rna. They used to be based in Boulder, and they moved to California now. They got bought out and shifted to California. And the purpose of this company was to design microRNAs to treat human afflictions. So you take a pill that had microRNAs, and the microRNAs would go in your body and help you what you need to accomplish. Some people call this, another word to describe these is microRNA. It's an M. Another word for the same thing is RNA interference. Does anyone know of any other terms for this? 3 different ways of referring to the same thing, okay? In general, I think I would say microRNAs are the actual RNAs. RNA interference is the process by which these things accomplish the interference. So, why would we evolve to have these microRNA systems to degrade things? Well, it's important. You've got feeding things in. Sometimes you need to degrade things. Hey, we're not experimenting, we're not, my cells are not in an environment where there's high lactose anymore, so we need to degrade those and move on and get some other RNA in there to be translated. It's important to have that. But more likely, this system evolved as part of our immune response. And I talked about HIV last time. And so the reason this lecture is after the alternate splicing is you understand that HIV genome is very dense. It's very small, but there's a whole bunch of genes, so the genes actually overlap, right? We talked about that one stretch that has 4 different proteins coded off of that one little stretch, right? So, in a virus, if we lay out a virus genome like this, the virus might make a messenger RNA that's this, from here, and it might make a different gene like this, and it might make a different gene like this. Oops. Right? So if I put all 3 of these in the cytoplasm, this guy can anneal here with this guy, and it can anneal in all of these spots with this guy, right? So then it would basically be like this. With these 2 hybrids, right? Make sense? So, I talked previously, what is ALU? Does anyone remember what the ALU sequence is?
It's a length of DNA that's repeated a lot of times.
It's a piece of repetitive genome. It's a 350 base pair sequence that is found in the human genome about 1 million times. If you guys remember back in lab, way back to computer work, and you would find your gene, and there were all these other genes on the chromosome that you could see. Do you remember that? You guys remember that image when you were in lab? Maybe blank stares for some of you. Yes. For those of you that remember, we'll continue. Right? So they kind of illustrated the chromosome like this, and there are all these dashes down here which showed all the different genes. In general, these genes did not overlap with each other. And there's junk DNA located between them. Junk DNA quite often would be repetitive sequences like ALU. What the effect of this is, in the human genome, one of the reasons only like 1% of the genome codes for protein, codes for something transcribed, 99% is junk DNA that when I was in school learned there's no use to it, it's just junk, just fills space. Maybe the reason we have that is so when we make transcripts, we don't have RNAs that overlap with that, that are hybridized. And as a general rule, in human cells, there is no double-stranded RNA. It does not exist. And this may have evolved so that when a cell does get double-stranded RNA, that is a sign that the cell must be infected with a virus. If evolution makes a definition, or some grand creator makes a definition, that human cells will not have double-stranded RNA, if a human cell gets double-stranded RNA in it, now it is not normal. There's a problem with that cell. It's infected with a virus. And the reason it's infected with a virus is the genomes of a virus have to be small, and the genes overlap and hybridize like that. So a human cell that evolves or is designed to have a mechanism that recognizes double-stranded RNA can then destroy that and protect itself from viral infections. So probably the system of finding double-stranded RNAs and cutting them apart and then using them to destroy other RNAs that match that sequence is probably part of our immune system. And it was probably later adapted for this, or maybe it was vice versa. I don't care which came first. Any questions on that? So one of you had a paragraph that talked about this panic response, PKR. Who had that paragraph? Does anyone remember? You guys did? So PKR is a protein kinase. And it's floating around the cell doing nothing, doing nothing, doing nothing. And it's looking, and it has a binding site for double-stranded RNA. If PKR binds to double-stranded RNA, PKR gets turned on, it tells the cell, we're infected with a virus, and sets up this panic response, this antiviral response. So, also, some viruses have double-stranded genomes. So when the genome gets dumped into the cell, it's double-stranded to start. It doesn't need to make any transcripts. PKR will bind to that and say, let's go. We've got a virus that's got an antivirus infection going on here. So, this enzyme that does the cutting is called dicer. And it was named dicer because it cuts things, right? Dices things up. It cuts up the double-stranded RNA, and then it loads the small bits into another enzyme. And that enzyme is called the Risc complex. So this must be German. Okay, it's with a K. Or maybe I'm going to impose communism. Everybody embrace it. So it's called the Risc Complex. It's a group of proteins. It's probably 6 to 10 proteins that all come together. And these, so dicer does the cutting and gets this small RNA here. Dicer then passes that small RNA to this other group. And dicer may actually be a part of this group. I can't remember now. And then the complex then takes the two strands apart. It may throw this one away, or it may throw this one away. To the best of my knowledge, it's random. And then it uses that as a guide RNA. And anywhere that Risc complex can hybridize, it will come in and it'll cut that RNA. So if it throws away the wrong strand, whoops, it's going around looking for nothing. It's not going to ever find a messenger RNA that matches that. If it keeps the correct strand, anytime it finds a messenger RNA that it can hybridize to, it will cut that messenger RNA in half. So if this is a messenger RNA that's got a cap and a poly A tail, when the Risc complex cuts that RNA in half, we have an RNA with a cap and no poly A tail. What happens to this guy?
It's degrading.
It gets degraded very rapidly. It does not have that protective poly A tail. This guy has a poly A tail but no cap. So this guy's going to get chewed apart from this side, and this guy's going to get chewed apart from this side. It rapidly goes into nucleotides. This is how you can have that double-stranded RNA, and it can perform an enzymatic function. Because once the Risc complex has cut the messenger RNA, it can then go off and use that same RNA as a guide RNA to find another messenger RNA. And to the best of my knowledge, what I've been reading is this complex can hold on to that little RNA for years.
It's a long time,
It's a long time.
especially for cellular stuff.
Yes.
So would it just like durate because the cell one cell left at a time would just sort of carry on mitosis like
So-
one other cell might get it, the other one might not.
How does this persist? First of all, these machines are very stable. They're not turned over very often. I have not read up on this in probably 5 years, so I'm getting out of date on this. But the last I was aware is when this machine cuts, it actually, once it cuts, it brings in a polymerase that makes a copy off of this. And now this is double-stranded RNA. And dicer will say, virus infection, panic. And dicer will come in and cut it, and now you can use all of that as new little guide RNAs that can be fed into the
So there's
system.
not just one rolling around with
Yeah.
a special little thing.
They can propagate their numbers.
Okay. That makes sense.
If you remember from the worm paper, they were injecting it into the head of the animals, and apparently those double-stranded RNAs were getting into the eggs, such that the progeny generation was knocked out for the genes. Probably happens in you. You say, oh crap, that's not good. Well, no, it's probably good because you're passing on immunity to viruses to your kids. Your kids are already equipped to deal with those viruses. Unless you were maybe immunized, didn't get exposed to the virus genomes, therefore didn't develop this response, then your kids would be susceptible to it.
But then we vaccinate the kids,
Oh, okay, then we vaccinate the kids. Good.
and everything's fine.
Kind of funny, isn't it?
Definitely say it works better than the alternative.
Yeah.
MicroRNAs are useful, but they're not the fail-safe.
Yes. Yeah, so the names of these are funny. Argonaut is one of the proteins in there. I don't know where they come up with these names. When I hear Argonaut, I think Argonaut liquors in Denver. You know, that's me. So it's a catalytic use, it's not a stoichiometric use, right? 1-for-1. Here's where the story gets even better. Sometimes Dicer, instead of loading it into the small RNAs, into the Risc Complex, R-I-S-C, it loads it into the Ritz Complex, R-I-T-S. And what Ritz does is once it gets loaded, is it goes into the nucleus. And it looks for that sequence. It uses the guide RNA and looks for a sequence on things that are currently being transcribed. So you've got your DNA currently being transcribed. You got a promoter, this, right? You got your +1. You got your transcript. You got RNA polymerase here with a transcript hanging off of it like this. And it's reading down until it's going to hit the stop right here. So the Risc complex tries to match and find the sequence in here. Oh, there it is. I've hybridized my guide RNA onto this. Risc then recruits histone deacetylases. What does the histone deacetylase do? Takes these subgroups off histones, which allows the positive charge of the histones to now operate. So when histones are positively charged, what do they do to DNA? They condense it. These histones also attract DNA methylases. So that what happens is the Risc complex recruits machinery that condenses this DNA. When we talked about protein or DNA chromatin spreading, this recruits the machinery that allows condensation of this and condensation of this. And the spreading, the condensation spreads all the way down on either side until it hits a boundary element. Such that if you've got your protein scaffold and the DNA is hanging off on it like this, if the Risc complex finds a gene in this rosette loop here, this radial loop, it will condense that in the whole radial loop. It will get highly compacted and not expressed. So it shuts down gene expression in both ways that they proposed in that microRNA paper. The Risc complex destroys messenger RNA in a catalytic process. But that's treating the symptoms of the problem. I'd be like, oh, I've got a headache because I've got a parasite infection in my head. I take aspirin to treat my headache, but you're not getting at the root of the problem. If I take a medicine that killed a parasite that's infecting the brain tissue, now I'm not going to get headaches anymore. So the system both treats the symptoms, and then it goes to the root of the problem, and it takes the DNA and sequesters the offending DNA away, potentially forever. Wow. Do you remember the other paper that the final two presenters talked about? Someone other than you two. What was that paper about?
About rice and the micro weird RNAs in the rice that get packaged and go through us and do stuff.
Rice appears as a transcript that doesn't code for anything. But when we eat that transcript, it survives the digestive system, it gets into our blood, and it's able to survive, travel through our blood, and get into our cells. And once it gets into our cells, it's used as a microRNA to modulate our cholesterol transport in our body. That's just one thing, rice. You think there's other things you eat that might affect your body? You guys are awful quiet. Is anyone blown away by this stuff?
It's kind of getting weird in this class, to be honest. It's a little sketchy.
It's sketchy. So I've said this before. I had a student who sat in the back one year and he's getting a PhD in biochemistry right now. And I think I've told you the story before. He walked in knowing that evolution was it. And it was after one of these lectures, he was a microRNA lecture or something. He walked up to me after class and said, now I know for sure there's a God. There's no way in hell that this evolved, just randomly from chemicals doing stuff. I don't care what you guys believe, but it's phenomenally complex, incredibly precise. It's very effective.
I'm sure we still don't even know half of it.
Yeah, we're still learning a lot. We know enough about it. So what this company, the siRNA company, siRNA company is they would get a sequence, and just like I was talking about the promoter bashing, is you have this sequence and we want to knock out that gene. What happens if I change nucleotide from a T to an A? What if I change nucleotide one from a T to a C, now a T to a G? So they would run through the entire thing, changing in every possible nucleotide, and then they would do combinations of those. And they would identify what is the best microRNA to knock out the whatever gene, the HIV genome in an infected cell. And so they're finding that there are sequences that appear to click into dicer and the RITs and the Risc machinery more effectively. So it's probably not random. There's probably specific sequences that these proteins are best evolved to handle.
And there's reasons for them.
Yeah, and there's reasons those proteins are shaped that way. Because it's all random evolution.
Well, it's random evolution that takes what works and keeps using it forever.
Yeah. Okay. Okay. Do you guys have any questions on that one? On microRNAs and how they operate?
Probably not ones that you could answer.
Ask them.
Well, I don't know how to say the question.
Yeah, I mean, if you want to go get a PhD, if this is interesting to you and you want to get a PhD, this is a great place. The Nobel Prize has already been awarded. You're not going to get the Nobel Prize, sorry. But, I mean, you could be the person who comes up with, oh yeah, just take this pill and it'll wipe out HIV from your body forever. Because if the HIV integrated into the genome, the Risc Complex will condense that to chromatin and it'll be lost away forever. And any virus genome that would be produced will be destroyed by the Risc Complex. Or, One other thing you want to knock out. You could design a double-stranded RNA.
We could cure herpes.
How about that?
Finally.
Finally cure herpes.
It only took us how long?
It's a funny story. I was in graduate school. Here's your commercial break for today. I was in graduate school. Does anyone know what HPV is? Human papillomavirus. It causes warts. There's probably 200 different versions of papillomavirus that infect humans. So the papillomavirus that affects the cervix of females, the bad ones are what, 16 and 18? Papillomavirus 16, papillomavirus 18. Like, you know, if a woman gets infected with those, her odds of getting cancer in her cervix is very high. Those viruses can't infect, you know, like the bottom of your foot and make a wart there. Each virus, each stage of the papillomavirus is specific for a location. While I was in graduate school, someone was developing a vaccine. You get a shot that would give you a vaccine and give you an immune response against HPV 16 and HPV 18. So the idea, which was really wild at the time, is that you could give someone a vaccine against the virus, and you could basically prevent that virus from infecting anyone in the human population, and it only affects humans. You could make that virus go extinct on the face of the earth, and the number, I think it was at the time, the number 2 or number 3 cancer killer of women, you could eliminate that type of cancer of women. Essentially 95% of cancers, cervical cancers of women, would be wiped out. So they were testing this vaccine, and of course, as nice as it would be to vaccinate a whole bunch of women and say, okay, you guys in the group that we're going to expose to this cancer-causing thing, and you are exposed to the cancer-causing thing, you know, who wants to sign up for that experiment, right? So you need to vaccinate thousands and thousands and thousands of women and then let them go live their normal lives, and then you measure who's getting cancer more frequently. The group that had gotten the vaccination had almost no infection, no cancer, and the group that got the placebo had normal levels of infection, normal levels of cancer. So the paper came out that this vaccine was actually working. And a professor came in to teach a class. The student raised their hand that morning and said, so I got a question for you. So you study HPV, and now that we've cured HPV, what are you going to do for the rest of your life? Right? So maybe it's not a good idea to cure herpes virus, because everybody's studying herpes virus and they'll be out of a job.
I don't know, and there's a whole lot of money in, you know, mediating and dealing with it.
There's a lot of money.
Pharmaceuticals lose a whole bunch of money. They don't like that.
Okay.
It sure helped normal people.
Let me try this on. I may regret doing that. Does anyone know what cystic fibrosis is? What is cystic fibrosis? You kind of vaguely know what it is. You can't describe it though.
Yeah, I don't know the proper words. Heard of it a lot.
Heard of it a lot?
Deals with nerves.
Deals with the nerves? No.
No?
It's multiple sclerosis.
Oh.
Cystic fibrosis. Lungs? Yes, it's lungs. What happens?
Fluid builds up in them?
The fluid builds up in them? Yep, that's
You
right.
can't like move mucus out of their lungs?
Yep, they accumulate mucus and they wind up suffocating. Yep. They die usually, if they make it to 20s, it's pretty amazing. So does anyone know the cause of most cases of cystic fibrosis?
Does it affect the cilia in your like?
Nope.
Apparently an autosomal recessive genetic disorder.
It's an autosomal recessive genetic disorder.
Yeah.
If you inherit a wild type copy and a mutant copy, you're a carrier and you're 12 by 5. If you inherit 2 mutant copies, then cystic fibrosis, my guess is you're dead around 20, 25-ish if you make it that long.
So it's just the time bomb. Genetic time bomb.
Here's the cause of it. So here's my protein coding region. There's a single nucleotide point mutation, such that this gene, which normally is going to be this long, starting with methionine, because all genes start with methionine, right? And ending with whatever the last amino acid is. This point mutation causes a premature termination codon. So you wind up getting a shorter protein. This protein doesn't work. This protein is a chloride channel. Allows chloride to go in and out of cells. So the reason mucus and liquid builds up is you're not properly moving ions to where they need to be. It's just that fluid accumulates in the lungs rather than in the body. Okay. What if I design a microRNA that was this sequence? And I took that microRNA and I put it in a little mister. And the person with cystic fibrosis would inhale the microRNA. What happens to their expression of this gene? It goes away.
And they don't have sodium channels.
Huh?
And they don't have like sodium channels at all.
Okay. So I know
specific.
if someone has on their chromosome, they have a mutant copy and a mutant copy, they're going to die by age 20. I give them this mister, but I'm killing them now, right? Right. By knocking it out, I haven't really helped the person at all. Now, what if I take a virus, and in that virus genome, I cut out part of the virus genome, and I put in a different sequence that I want. If I put in the wild type cystic fibrosis gene, and I put that virus, we'll call it adenovirus, in a mister, person who's now knocked out this bad chloride channel, and they inhale the mister, they get the virus infects all their cells, dumps the genome of the virus, including the wild type version of cystic fibrosis, into the cells. Now, it's amazing, and every cell in the lung gets this gene. Is the cell, have I complemented the mutation? Have I rescued the cell? I see some yeses, and I'm going to say no, why not?
microRNA is going to kill the wild type.
Oh,
The microRNA is going to kill the wild type. The microRNA matches this right here, right?
Now we have to figure out how to kill the killer one. Or just kill her.
But if I get rid of the microRNA, I'll get the mutant version.
But because
Oh, but I have a wild type.
happen, there's also a wild type. But
So
also,
now what I've got, yeah, what you're saying is now I've got it.
all the way.
Now I've got what the carrier.
Couldn't you make it go all the way down to the DNA though and get it to?
Yeah, but then it'll shut this off too.
It's complicated.
Think about the genetic code of DNA. Anyone think of a solution that I can make this work?
You make microRNA for something somewhere else on the radio loop for the cystic fibrosis that they have that's not on the virus.
There's a solution. And consider that brilliant. I make a microRNA not for the gene, make a microRNA for some other gene on that same radial loop. The Risc Complex will condense all of it and I'll knock this guy out. But when I put this guy in, it won't get condensed. Because the microRNA doesn't target that RNA, it might target some other RNA. Okay. Awesome, I didn't consider that. Some other solution.
Do people actually like try stuff like that?
Yes. And it killed a little boy in Pennsylvania, so they stopped the trials. Despite a lot of people being helped.
Do you know why it killed the little boy?
The kid got too high of a dose of the adenovirus delivering the wild type, and he had an allergic reaction to that high dose and he died.
But does it help other people?
Immensely. The people who would take the virus, their lungs cleared up in a couple of days, and they're like, oh my gosh, I've never been like this before.
Then they had to stop taking that medication
The
anyway?
US government shut it down, said you may not deliver viruses into people.
That's exactly why we need to keep doing the stuff so we don't do that anymore.
Can someone think of a solution? Tell me about the genetic code. Remember this? Then I guess we go C-A-G-T. What am I drawing here? Oh, that protein list. This is the translation table, right? So are there 64 different amino acids? There's 64 codons. How many amino acids? 20. What does that mean?
Lots of redundancy.
Lots of redundancy.
Change one letter, you get the same thing.
I could change one letter, but still get the same amino acid put in there.
Or not, depends, but you can.
Anyone see where I'm leading you on this?
You change it enough and the microRNA will bind to it, but you still come up with the same exact code.
I'm not going to put wild type sequencing here. I'm going to
Put
put.
special fancy type.
I'm going to dramatically change the sequence of this. But I'm going to make sure the exact same amino acids get added. Does that make sense? Such that the microRNA can't recognize this.
Whoa.
Because that's what they actually did.
And it works. How cool is that?
Too bad the government's a bunch of boneheads.
They kind of loosen this up, and they're doing some virus testing nowadays. But it was shut down for 5 years, 10 years.
So is that the standard medicine that kids with cystic fibrosis can take now?
To the best of my knowledge, it is not a standard medicine, but I have not followed that story in a while. But potentially, someone with sickle cell anemia, it's the same thing. It's a change in one single nucleotide. So if you could just knock out the wild type version, and then infect them with a virus that has wild type-ish, all those, yeah. This modified code version, but it's going to create wild type amino acids to make a wild type protein. Any gene like that. We could cure my hitchhiker's thumb, right? No, because that was development. I was probably stuck in that situation. Okay. You guys have comments or questions? Yeah.
Have you put the PowerPoints online?
Most of them. I do know. Let's look and see.
Yeah, I do not believe they're all on.
There's 4 of them.
To the best of my knowledge.
1, 2, 3, 4, 5, 6, 7, 8 lectures so far.
And that's the whole.
Oh, I don't have the ones that Josh did do I?
Yeah.
I need to put those up. Okay, will do. All right, thank you.
No.